right. So let's get started when other people will be joining us. Welcome everyone to the International Hooper Education and Training Committee webinar series. The Education and Training Committee at Hooper facilitates collaborations, supports the organization of the annual World Congress, and organizes programs such as workshops, courses, and tutorials that introduce novel technologies and promote the professional growth of Hooper members. The 2024 Hooper World Congress will be held in Dresden, Germany in October 2024. Hope to see some of you there. Today's seminar marks the third session of this year, and this series is our second webinar series hosted by Hooper ETC. I'm Shisha Huang, a professor and director of the antibody-based proteomics core at Baylor College of Medicine, Houston, Texas. I'm the chair of today's webinar with co-chair Dr. Brandin Chazarin. Brandin?
Hi attendees, hi everyone. I'm Blandine Chazarin. I'm a postdoc in Prism Lab in France, and I specialize myself in single-cell proteomics.
Okay, we are honored to introduce our speaker today, Dr. Isela Gallagher, a senior research scientist in George Mason University in Fairfax, Virginia. Dr. Gallagher has accumulated more than 15 years of expertise in reverse phase protein array and laser-captured microdissection technologies in the Center for Applied Proteomics and Molecular Medicine, led by Drs. Lance Liotta and Emilio Petricoin, the inventors of both technologies. Dr. Gallagher's current research focuses on the molecular profiling of tumor cells, utilizing LCM and RPPA to investigate signaling pathways and new disease mechanisms. In addition to her research projects and training graduate students and postdocs in the lab, Dr. Gallagher has been devoted to training high school and college students. The training program initially initiated within their own groups and now organized by the university is called the Aspiring Scientists Summer Internship Program. This program places close to 100 students each year in research labs during the summer. Please remember that this webinar is being recorded, so any questions you ask during the Q&A session will also be recorded. If you would not like to be recorded, you could turn off your camera during Q&A. And Dr. Gallagher, we're excited to hearing your seminar today.
Excellent. Thank you, Shisha. Let me share my presentation and make sure that everything is ready to go.
Yes.
Can you see my presentation?
Looks perfect.
Excellent. Well, thank you, Shisha, for the lovely introduction, and thank you to the ETC as well. I appreciate the invitation to speak here today about reverse phase protein arrays and some of the very important applications of this platform, especially in cancer research. I am told that I have about 45 minutes, and afterwards there will be some time dedicated for questions. So let's get started. The title for my presentation today is Reverse Phase Protein Array Applications, Molecular Profiling, and Predicting Response to Targeted Therapies. I'm sharing with you the agenda for this presentation. In the interest of following with the ETC focus on education, I will give you an introduction to reverse phase protein arrays, RPPA for short, the workflow for this technology, and we'll also introduce laser capture microdissection, which is another technology that our laboratory utilizes in combination with RPPA. And then I will also tell you about some important research that our lab has recently completed to give you some real clinical examples of RPPA applications. RPPA is a technology that has the ability to generate a functional map of known cell signaling networks or pathways. It is used to quantify proteins and proteins post-translational modification in cellular lysates and also body fluids. RPPA, or reverse-phase protein arrays, have evolved over the past years as an innovative, nowadays well-established and accepted protein profiling platform to study protein expression, levels, and activation status of cells signaling pathways and networks. So the microarray platform combines high-throughput analysis with minimal sample consumption due to its miniaturized format, and has proven its value in translational research, particularly in the field of oncology. So as demand for patient-tailored therapy increases, so does the need for precise and sensitive technology to accurately profile the molecular circuitry driving an individual patient's disease. And currently, RPPA has been able to meet that demand. In the last decade, the field of molecular medicine has seen new technological advances in proteomics and genomics. Genomics, as we know, is the information archive. However, the proteins that are the ones that do the work of the cell. In most cases, the drug targets themselves, actually for almost 95% of the FDA-approved therapies of every disease type, proteins are the targets themselves. At the genetic level, certain chromosomal deletions, rearrangements, and gene mutations are selected out during cancer progressions because these defects ultimately alter protein signaling networks and generate a survival advantage for the cell, especially the cancer cell. However, the function of proteins is closely tied to their cellular tissue and physiological context and the protein. So protein interactions that drive biological outcomes can be characterized as fluctuating information flow within the cell. So defective or hyperactive or dominating signal pathways drive cancer growth, also survival, invasion, and metastasis. So reverse-phase protein microarrays are one of the tools available for profiling molecular pathways in a given cellular sample. This type of microarray can uniquely quantify phosphorylation or activation states of protein. So, as I mentioned, RPPA is a higher throughput, lower cost procedure compared to other technologies like mass spectrometry, and is therefore more approachable to rapid analysis of large numbers of experimental samples. So in this slide, we have the technology itself. It was invented by combining the fundamental concepts of protein lysate transfer to nitrocellulose membranes and miniature assays with robotic printing technology of small amounts of tissue. The term reverse-phase protein array was first coined in around 2001. In the first paper describing the technology, pioneered by Dr. Lance Laiota and Dr. Emmanuel Petricoin, the term reverse phase refers to the fact that the analyte is immobilized as a capture molecule, rather than immobilizing an antibody as the capture molecule itself. So each RPPA microarray consists of a self-contained assay comprised of either duplicate or triplicate samples. We can also add controls and calibrators that are analyzed with one class of antibody and also an amplification chemistry. So the first RPPA was used to construct the first quantitative multiplex cell signaling protein analysis of tissue cells. In this experimental application of RPPA, the technology was used actually to discover the importance of the pro-survival pathway during the transition from in situ to invasive prostate cancer. So this technology provided the sensitivity and precision to measure phosphorylated signal proteins in small samples that were microdissected directly from the invading edge of the tumor. Since 2001, when this first paper came out, in the literature, we have seen other names that to refer to reverse phase microarrays like lysate array, reverse phase lysate microarray, protein microarray. So thanks to the RPPA Society, it was agreed and suggested that we stick to reverse phase protein arrays. So the term RPPA is therefore the one that we're using currently. So RPPAs are applicable for profiling key cellular signaling pathways and protein networks, allowing the direct comparison of the activation state of proteins from multiple samples within the same array. The nature of cellular lysates, either tissue material or others, are spotted directly onto nitrocellulose coated slide, and multiple samples are simultaneously probed with the same antibody. Each sample may be printed in either a statewide dilution curve with either using colorimetric or fluorescent detection. Similar to an ELISA or immunoassay, high and low controls and calibrators can be printed on every slide with the RPPA format. Of course, to ensure inter and intra-assay reproducibility, and also check for QA, QC, and fidelity of data generated as well. So the final values for any patient sample, for example, can be interpolated to the reference calibrator printed on every array. Multiple samples can be printed on the array, as I mentioned. This is a high throughput approach that can test and quantify about close to 1,000 samples on a single slide. It has high sensitivity. It's applicable to very small sample sizes and less than 10 cell equivalents that you can detect here. It detects and quantifies hundreds of different proteins at their expression levels and modification status in about 80 microliters of cell lysates. It can be quantitatable. Serial diluted samples enable detection of proteins in their linear range to reliably determine relative protein levels of samples printed on the array. So I'm gonna talk about a little bit about laser capture microdissection, because it's a technique that allows the isolation of pure cell populations from a heterogeneous tissue section. You can, under direct microscopy, you can visualize the cells. And since a tissue sample can have, it's very heterogeneous, a common problem encountered by both genomic and proteomic researchers during tissue analysis is that you have this heterogeneous tissue. So previous proteomic studies relied on whole tissue lysates derived from samples with uncontrolled cellular input, including mixtures of stroma, immune cells, and tumor epithelial, along with fat and fibroblasts, etc. And it didn't adequately enrich for any cell type greater than 90%, which we have demonstrated greatly impacts the accuracy of protein and protein signal data. So the molecular profiling of pure cell populations, for example, tumor cell populations, which is reflective of the cell populations in vivo genomic and proteomic state, is essential for correlating molecular signatures in normal and diseased tissue. And this technology was developed by Dr. Laiota and when he was at the NIH. And it uses a thermolyable polymer, which is placed directly on top of a tissue section on a glass slide. This can be either a frozen tissue section, around 8 micron thick section, or FFPE tissue as well. An infrared laser melts the polymer in the vicinity of the selected cells. And then this polymer melts on top of the tissue. And once you lift the cap, it removes the tissue with it. So the melted polymer, as you can see on the panel on the top, you have an effective microdissection if you can see a circle that properly melts onto the slide. So if you have inadequate power or duration settings, it creates like a fuzzy looking spot, and this lacks a distinct black ring or a dark area in the center of the spots, as you can see on the slide. So a section of the tissue is mounted on and stained on a glass slide, imbued under high magnification. So the cells of interest are selected, then lifted out of the tissue by the action of a laser pulse. The infrared laser then locally expands the polymer, and then you can capture the tumor population or the cell population that you're interested on. So when the film is lifted from the tissue section, it only selects the cells that you actually selected. After that, protein extraction is very easily using extraction buffer directly on the cap, and the pipette is usable, and you can deliver the extraction buffer on top of the cap. And this you collect the extraction buffer, and it contains the solubilized cells that you need to then print onto the RPPA. So previous proteomic studies relied on whole tissue lysate derived from samples with uncontrolled input, including mixtures of stroma, immune cells, etc. So with LCM, we have demonstrated that we can enrich, for example, the tumor population for RPPA analysis. So the laser capture microdissection reverse phase protein microarray workflow, it's presented here. We usually get the clinical tissue samples, and those are subjected to laser capture microdissection. We can either, as I mentioned, receive frozen biopsies or FFPE samples, and we can work with any type of tissue, and we can procure highly enriched and purified cell populations that are then lysed and printed onto nitrocellular slides. Each slide is then exposed to a specific primary antibody and then secondary antibody that amplifies the analyte concentration within each spot to produce a colorimetric or fluorescence signal. Then it is then captured by the appropriate detector type and analyzed. So genetics is already being used to assist clinical decisions making in specific cases, but additional levels of biological information are required to better understand disease and more accurately predict phenotype from genotype. So a crucial source of information in this context is the proteome and the activation status of dynamic cell signaling pathways through post-translational protein modifications. So nowadays we have an arsenal of molecular targeted therapies, many of which are tyrosine kinase inhibitors. These molecular inhibitors won't fulfill the promise unless therapy is tailored to individual patients' disease. And thus we refer to personalized therapy. Importantly, as we know, most current therapeutics are directed at protein targets, and these targets are often cell surface receptors, protein signaling molecules regulating different pathways, such as apoptosis and growth, or protein kinases and other substrates. So in the era of personalized medicine and targeted cancer therapies, identifying patients that will benefit from existing and new therapies is critical. So RPPA, it's able to measure the abundance and activation of the cell surface receptor proteins and their downstream signaling pathway. So these biomolecules serve as the drug targets for most FDA-approved and investigational therapies for cancer. So RPPA may aid in determining the ideal prescribed medication or for patients based on the unique molecular characteristics of their tumor. Up to this time, RPPA technology has graduated from a research tool to an essential component of clinical drug discovery research and personalized medicine. Although it was initially designed for identification of alterations in protein signaling pathways in cancer, it is easily adaptable for assessment of protein pathways in other diseases and cell biology processes. And this slide just contains a few examples of manuscripts that have been published lately. And the count is going up for RPPA. So we hope that many more people gets introduced to RPPA and learns how to take advantage of this technology. So RPPA technology generates information regarding the phosphorylated state of key signaling molecules and the configuration of tumor cell signaling networks that is not possible to obtain by genomic profiling. So patient clinical information combined with protein network data obtained from RPPA can provide insights into mechanisms of receptor blockade, for example, or compensatory post-treatment receptor activation and overall differences in pathway signaling between patients and even between treatment arms. So the results of which pathways are in use or activated can then be correlated with biological and clinical information and serve as both a diagnostic and a therapeutic guide. This technology is currently utilized in clinical trials for profiling and comparing the functional state of protein signaling pathways, either temporally within tumors, between patients, or within the same patients before and after treatment. And so I'm gonna show you in a bit with the two examples that I have here to present on the clinical trials that we have used RPPA. And so the growing awareness that every cancer develops as a unique molecular entity depending on the patient's individual genetic background is leading the development of personalized therapy. Although two tumors could appear morphologically similar under the microscope, they could actually be driven by different aberrant signaling pathways, or for this reason, a population of cancer patients treated with the same drug may result in only a few responders, and many others could suffer from unnecessary toxicity. RPPA analysis quantifies specific phosphorylation events and offers an approach to profile the activity state of protein cascades and that contain potential drug targets to segregate responders from not responders, for example. So each array is probed with only one antibody. Replicate slides are printed to probe for approximately 300 different antibodies that we have in our current inventory to produce a functional proteomic profile of each sample. So the RPPA data provides relative quantification of phosphorylated or glycosylated, even acetylated, cleave, and total cellular proteins in disease or non-disease tissue from multiple samples printed on a series of identical arrays. These arrays can be measured and compared in parallel using commercially available antiphosphoproteins or other specific antibodies. Which brings me to this point, because RPPA technologies is highly dependent on the quality of antibodies, in antibody specificity has to be evaluated and validated beforehand using conventional immunoblotting techniques such as Western blotting or a broad panel of cell lines in human tissues to validate it before we can use it in our RPPA arrays. So we have to see a single or dominant band at the appropriate molecular weight. In this way, we can validate the specificity of the antibody that we can use. So the RPPA has the ability to generate a functional map of known cell signaling networks for an individual patient obtained directly from a small biopsy. The high sensitivity and good reproducibility of this technology has motivated its application to a wide range of sample types, including cell lines, preclinical, or, for example, xenograft models, and patient-derived material. Indeed, the microscale printing of very small amounts of samples is of particular benefit for analysis of limited clinical or preclinical important material. And fortunately, RPPA has become a powerful addition to the biomedical analytical toolbox for the investigation of disease mechanisms, diagnosis, and prognosis as well. So diseases such as cancer, we know that they are caused by dearrangements in cellular protein molecular networks. These pathways contain a large and growing collection of drug targets governing cell survival, proliferation, invasion, and cell death, for example. The clinical utility of RPPA lies in its ability to generate a functional map of known cell signaling networks from any obtained biopsy or specimen that you would like to investigate. To give you an example of how RPPA is being used in the clinical setting, I'm going to tell you a little bit about the work that we did as part of the Fairline trial in collaboration with investigators from Genentech. One of the objectives of the Fairlane clinical trial was to address the need for more effective therapeutic approaches with appropriate diagnostic biomarkers for triple-negative breast cancers. We know that triple negatives are very aggressive cancers with poor prognosis and high recurrence and metastatic rates. So due to the molecular heterogeneity of triple-negative breast cancers, a key aspect for targeted therapy is identifying tumors that are most likely to be sensitive to the specific oncogenic signaling perturbation to maximize the clinical benefit. So in this trial, patients with early triple-negative breast cancers were randomized to receive either paclitaxel with ipatasertib or paclitaxel plus placebo for 12 weeks prior to surgery. Our group performed the RPPA analysis for cell signaling proteins from matching frozen tumor samples collected either at baseline and at cycle 1, which is around day 8, to characterize AKT signaling activity and its association with other genomic or IHC-based PI3 kinase AKT pathway biomarkers, and also to investigate the activity of AKT inhibitor ipatasertib, which actually has shown great efficacy in patients with metastatic triple-negative breast cancers. In this trial, we found that the AKT and mTOR downstream components clustered tightly together, showing here the pairwise correlation between all endpoints measured by RPPA at baseline. We see that all major AKT signaling components measured, including AKT and its known kinase substrates, clustered together in the same manner. All the components of the mTOR kinase and its own substrate cluster together in another group, as you can see on the squares, orange and green here. So as we see in the panel below, AKT and mTOR are often depicted in the linear PI3K AKT mTOR pathway. The activity of mTOR is actually modulated by several other signaling inputs in addition to AKT. So this kind of explains the two clusters that we saw at baseline. So the PIK3CA AKT PTEN alterations, together with multiple cell signaling activities, modulate the level of phosphorylated AKT on the phosphorylation site, serine 473 and 309, 308. Importantly, we found that tumors with high phosphorylated AKT levels exhibit the strongest association with enriched ipatasertib activity, suggesting that phosphorylated AKT tumors are most addicted to AKT signaling. So this particular study provides proof of concept that the baseline phosphorylation levels of AKT, which is the direct target of ipatasertib, could have predictive value and may possess an improved means of biomarker-based patient selection for AKT inhibitors and diagnostic utility for precision medicine. In this slide, we're showing the PCR in the IPAT plus paclitaxel versus placebo plus paclitaxel arms among different biomarkers selected patients or populations. We observe an increase in PCR among the phosphorylated AKT and mTOR subgroups. We then examined tumor overall response rate by MRI and found that the phosphorylated AKT high but not mTOR show a significantly higher overall response rate with ipatassertib. We also found increased overall response rate with ipatassertib regardless of the next generation sequencing or IHC biomarker status investigated here. This results suggest that tumors with PIK3CA, AKT, PTEN alterations, either by NGS or IHC, are indeed associated with higher levels of AKT protein phosphorylation, indicative of AKT activation. So the genetics is already being used to assist clinical decision making in specific cases, but additional levels of biological information are required to better understand disease and more accurately predict phenotype, such as levels of phosphorylated AKT from genotype. A crucial source of information in this context is the proteome, and notably the activation status of dynamic cell signaling pathways through post-translational protein modifications. So indeed, genomic mutations are not always associated with activated signaling pathways, and conversely, pathway activation can occur in the absence of mutations, as exemplified by the PIK3CA and HER2 signaling in breast cancer. Another excellent example that utilizes RPPA, it's the I-SPY 2 trial, which is another trial that our center is very excited to be a part of. This is a multi-center phase 2 trial that uses response adaptive randomization within biomarker subtypes to evaluate a series of novel agents when added to standard neoadjuvant therapy. And this is for women with high risk stage 2-3 breast cancer. So within each patient subtype, participants are assigned to one of several investigational therapies or control regimens. And randomization probabilities are proportional to current probabilities that the respective therapies have a higher PCR rate than control rate in the respective subtype. So for this particular trial, the primary endpoint is pathological complete response. And we're looking for no residual disease in breast or nodes at surgery. The goal is to identify and graduate regimens that have that are equal or more to 85% predicted probability of success in a 300-patient phase 3 neoadjuvant trial defined by hormone receptor and HER2 status and MammaPrint. So the regimens may leave the trial for one of 4 reasons. They can graduate, they can drop for futility, they can be dropped for safety issues or occurring maximum sample size that is needed for the trial. So one of the objectives is to further advance the ability to practice personalized medicine by learning which new drug agent are most effective with which types of breast cancer tumors. And another one is to identify tissue and blood biomarkers that correlate and predict responses to each therapy. And so we can learn more about which early indicators of response are predictors of treatment success. So it is a huge undertaking and it involves multiple centers. And up to this point, I believe it's about 2,200 patients so far. So it is a big clinical trial. We recently published our results in Cell Reports Medicine. We completed the largest proteomic-based drug target activation mapping that encompasses more than 700 breast tumors. And these 700 breast tumors were microdissected. So they were tumor enriched, and in this study, we identified phosphoproteins and protein-based signatures of global resistance, and treatment-specific and response predictive signatures. And all of this was done in pre-treatment tumors. So we also found the HER2 EGFR protein activation signature that could be used along with transcriptomic-based subtyping to provide better outcomes in triple-negative patients. So the I-SPY series of clinical research studies is truly a great example of how RPPA can be used to discover novel tissue cellular signal pathway protein biomarkers that are predictive of therapeutic success for patients undergoing therapy with experimental agents. So I-SPY, the study identified 3 specific proteins activated and expressed in human breast cancer that didn't respond to any treatment, no treatment at all. The identified biomarkers associated with global resistance are cyclin D1, estrogen receptor alpha, and phosphorylated androgen receptor. Some of the resistant markers, for example, cyclin D1 expression associates with resistance in all subtypes looked at in this study, except HR negative, HER2 positive subtype. Estrogen receptor and androgen receptor expression levels associated with resistant overall in the HR positive, HER2 negative subtype. We also identify some sensitivity markers, for example, STAT1, phosphorylated STAT1 and phosphostat5, which are associated with treatment sensitivity in all subtypes. Phosphorylated HER2 1248 and EGFR and phosphorylated EGFR also associated with treatment sensitivity in HER2 positive patients. And this was very important, and I will mention it in a couple of slides more. Something about this signature that was identified in this set of samples. Here, this one way hierarchical clustering map of proteins and phosphoproteins shows basically the mean signaling within each RPPA signaling clusters. This study set provided 11 protein signaling pathway-based clusters comprised of patients with common signaling activation patterns and reveal druggable targets that may not have been the target of the therapy they were randomized to receive in the trial, and also identify specific precision therapies that will be optimized for best response. For example, the patients in clusters other than 7B, which 7B, by the way, has the highest PCR of all the clusters, generally were not provided therapies that match the underpinning pathway activation signatures measure. So we identified patients in cluster 1, for example, with elevated immune-related signatures that could have possibly benefited from immunotherapy agents instead. Another example is cluster 5, which was characterized by DRD pathway activation, also elevated AKT phosphorylation and Tie2 expression, which suggests more patients in this cluster might have achieved PCR by including AKT inhibitors in their treatment regimen, in addition to immunotherapy and or Tie2 inhibitors. So it was a great way to see if there were other venues that we can explore for this particular patients. So we introduced a dichotomous HER2 activation response predictive signature that we're calling HARPS. For stratifying triple-negative breast cancer patients to either HER2 or immune checkpoint therapy response as a model for how protein activation signatures provide a different lens to view the molecular landscape of breast cancer and kind of synergize with transcriptomic defined signatures already in place. This two-way scatter plot of HER2 phosphorylated at 1248 in the x-axis and EGFR at 1173 of triple-negative breast cancer patient populations with predefined neratinib responses cut points that we use to determine the HARPS positive tumors. The bars here represent response rates observed for triple-negative breast cancer patients whose tumors were HARPS positive in blue or HARPS negative in yellow across the 6 treatments arms for triple-negative breast cancer patients. So the triple-negative transcriptomic signature distribution in both the HARPS positive and HARPS negative cohorts can be seen on the lower panel. Why is this important? So we know from past work that HARPS positive triple-negative patients are exceptional responders to the dual kinase inhibitor neratinib compared to HARPS negative triple-negative patients. So taken together, these results strongly suggest that the co-activation of these two proteins, HER2 and EGFR, gives us a way to identify HARPS positive patients in triple-negative tumors, that these patients transmit a biological relevant and functional signal in vivo that the HARPS signature accurately predicts therapeutic response to a precision therapeutic modality. So the use of HARPS in triple-negative breast cancer could actually help and result in about like an 80% response rate compared to the current 30% to 40% response rate. So it is, we're very hopeful that this can be validated in a different setting and to prove this concept. So because we found that global resistance to all drugs tested in the study was based on these 3 proteins being elevated or activated, we can actually target or turn off these proteins directly, which will help the I-SPY trial clinicians and their pharmaceutical partners prioritize new therapeutic strategies that target these proteins. The results give us clear targets that we can use to identify promising agents and test them in the I-SPY 2.2 trial, which is coming up. And we can see how the bench to bedside translation is happening while we are coming out with results from this trial. These findings are very important as inclusion of new androgen receptor and ER targeting endocrine-based approaches could be rationally considered in prospective I-SPY2 arms to address resistance. So our results showed that developing new treatment approaches for patients whose tumors display this characteristic protein signaling signature, as well as others with global resistance phenotype, it's very urgent. Our ability to analyze the RPPA generated protein signaling data across arms, agents, and subtypes provides a real important opportunity to analyze and uncover protein expression and signaling activation biomarkers and signatures from a large pool of patient tumor samples that associate with global non-response or resistance, and as well as indicate possible new therapeutic targets and strategies for therapeutic prioritization. So in summary, from a single biopsy specimen, the activation or in-use state of over 200 proteins, drug targets can be quantitatively measured at once, providing unprecedented opportunity to measure the direct targets of nearly every FDA clear and experimental targeted inhibitor at once from just a small piece of a single biopsy sample. I want to acknowledge and thank our collaborators from both the I-SPY2 trial and the Fairlane trial for the opportunity to work with them, and especially our hardworking George Mason University team that it's very supportive and always open to new collaborations. Thank you very much for your attention and for letting me share some of the RPPA work with you. If you have any questions, I'm here to try to answer them. Thank you very much.
Thank you for this presentation. It was very interesting. I have to confess that I heard about this technology, but I was definitely not aware of what you can do with it. So we got a question regarding the detection of phosphorylation on FFP tissues, but like very old FFP tissues. Do you have any limit in the time to use a FFP tissue? Is there any results that can be biased by the fact the FFP tissue was stored for a long time?
That's an excellent question. We actually can attempt to analyze and use RPPA with any FFPE tissue. However, we won't know until we analyze the data. So we would probably attempt to print and microdissect this old tissue and also print controls and calibrators that can tell us or give us an idea if the data it's reliable and true to its results as well.
And is there any recommendation you may have about the storage of this kind of samples to facilitate the analysis with your technology?
Yes, we actually store most of our FFPE samples at 4 degrees with in plastic bags with desiccant. So it has, at least in our laboratory with our experience, it has helped us preserve the tissue a little bit longer and keep the feasibility of those tissues active.
Okay. And so I think my next question would be, so you compare this technology to ELISA, for example, or blotting technologies, right? How much would it cost to do a patient sample on a classic panel like the ones you present, is it something affordable? Is it something that the cost could be a limitation for the, you know, the move to the clinic?
I'm not particularly sure about the cost at the moment. I know it's now commercially available, the assay through Theralink Technologies, which is a spin-off company from that came out of George Mason, our laboratory. So I can give you definitely their information and see about the cost of a particular sample. I'm not familiar exactly with the actual cost, but it is available at the moment through Theralink.
Okay, thank you.
I would like to.
Yeah, I was
I would
saying
like to add something about the cost, since I have a core facility actually doing the reverse phase protein array. So the actual cost, it's pretty high because of the antibodies, the reagents, the instrument, the timing. I think the chat has another question, and as I will answer later on. But for us as a core facility, because we could also obtain grants. So like for my lab right now, we have a grant for the last 15 years, for 15 years. So they charge back for the researchers. If it's a clinical project, so the company does the clinical project, that's amazing. So they are clear certified everything. So if it's a research project, my lab will be able to do that with the cost. Currently the cost is $149 per sample for the academic price, because we are heavily subsidized by our grant. But the real price is at least 4 times higher than this.
okay, good to know.
Back to you to the questions. I think that's a chat question.
Yeah, exactly. So we have one more question from the chat. How many proteins are printed on each slide, and which PTMs are currently available to be screened by this technology?
Okay, so we can print up to close to 900 spots. So it depends on how you want to construct your array. So we usually in our lab print every sample in triplicates. So that reduces the number available to print in one array. So it depends on your own needs and how you want to analyze the data for a particular study set that you have. But up to 900 spots can be printed in one of the arrays, avoiding, of course, like the very edges of the pad, and to avoid any staining issues and scanning issues. That also reduces a little bit the number of samples that you can print, but it is a large number compared to other technologies. And we have in our laboratory close to 300 validated antibodies that we can probe these arrays with. So if we have a large study of samples, for example, the clinical trials that we collaborate in, we can probe all those 900 spots with one antibody at the time. So you can have information readily available for you for that particular protein. So.
And how long is it taking you to start with the sample until you get the results ready for interpretation?
So again, it depends on how many samples are in your study. So if you have a small set of samples, and it depends whether or not you want to couple your RPPA with LCM, the laser capture microdissection for enriching tumor cells, for example, that it's a little bit more time consuming, but the actual staining and analysis of the RPPA, it's a little bit more quicker. And so it really depends on how many samples you have on your array. And but the analysis and scanning of the array can be pretty speedy.
Okay, good to know.
I have a follow-up question on that. And how many, what's the amount of protein you could achieve after laser captured microdissection per sample?
So if you have a core biopsy, for example, we usually ask to either for the collaborators to send us from 5 to 10 slides, either frozen or FFPE, to evaluate how much material we need. So if the tissue sample is big, then and it has a lot of tumor, we can microdissect the minimum of, I will say, like 2,000 to 3,000 shots. So it will be small volume. For example, a lysate of maybe 10 microliters, you can print up to 100 arrays. We want to achieve more like the 20 microliter lysate that will yield, for example, up to 200, 250 arrays for that material. And as far as the protein concentration of the material, we would like it at least to be like 0.25 micrograms per microliter in order to have a good data and for analysis.
Oh, wow, that's quite a lot from the microdissection. So how long does it take to finish one sample for microdissection at that level?
So if, for example, we can, if we are microdissecting, for example, 5 slides to achieve around 3,000 shots or per sample, it will be maybe half a day to microdissect. If we need more slides, or if the sample it has very little tumor, we might take the whole day to microdissect. But then once you have the lysate, printing the lysate takes not very long, and it just depends on how many arrays you're printing. So if you are printing around, for example, 10 arrays, that can be done in 2 hours. And the staining of it can take up to 4 hours, and then analysis. So everything can be done in I would say 4 or 5 days.
That's fantastic. It looks like if you half day or whole day for one sample, you had 700 plus samples for your I-SPY trial. That
Yes.
takes 2 years to get all the
Yes.
samples, then a week to get the RPPA data.
Yes, it was quite the undertaking for this I-SPY trial, but it is so important to do, and the results have helped so many patients already. So we're proud to be part of that clinical trial.
Absolutely. So Isela, many of this group and a lot will be watching the video later are
Yep.
from the mass spec community. So have your study been done in comparison or use both technology after microdissection, or could you comment on that?
Can you
Like using mass spec and RPPA, or using either technology? Since my lab has, for some of the studies in my lab, we used both technology. We found it really complementary, because a lot of people are saying that, okay, if I would like to do discovery based, I should go to mass spec. If I want to go to target based, we go to RPPA. But in my hands, I found out actually RPPA could be also discovery based, because for the about 300 antibodies, a lot of labs, they do not know what's the involvement of these 300 proteins, antibodies. And my colleagues doing mass spec, at least Brandon, you probably know more about that. For profiling phosphorylated protein by mass spec, it's still quite challenging. You need a specialized enrichment in advance, right?
Yeah, and with the RPPA, you don't need any fractionation or anything else for your samples, so you can print them as they are, and you can probe with our panel of available and validated proteins, and approach the biomarker discovery that way. With mass spectrometry, it is still usable, and we do have a couple of mass specs here in the laboratory, and we use also RPPA to validate sometimes results from mass spec. So it definitely can be a complementary approach for this. But if you want to measure the activation and the phosphorylation of patient samples, RPPA, I feel it is a faster approach and more, you can tell more that is approachable to the biological work that the tumor is doing, or the particular cell is doing, if I'm making myself clear.
Yeah, absolutely. That's a great point. So the laser captured microdissection is really great in identifying homogeneous samples, right? Do you still have some heterogeneity using microdissection, or the problem is taken care of?
So there may be some contamination of other cells, but we think that we can enrich the population of cells up to around 90%. So it is representative of what for example, the tumor cell population is giving us as far as activation or phosphorylation of the tumor. We do acknowledge that there may be some carryover or contamination when you try to get those tumor cells that are in areas with a lot of connective tissue or lymphocytes or fat cells, but that's why we call it enriched lysate. But there may be some residual from neighboring cell populations, but not as, it doesn't impact the overall result as much as previous studies where you use the whole lysate to try to explain the activation or profile proteins in a particular sample.
Yeah, that's fantastic. Yeah, the laser capture microdissection is so powerful. The only is that not all the lab could do that, right? It's quite technically quite challenging to do. So my lab does whole cell lysate, whole tumor lysates. So that's
Okay.
much faster, and also it's pretty powerful as well. So I'm
Yes.
thinking, yeah, I'm thinking in terms, I think I'm directing a question to Brandine. So for RPPA, the normalization. So first of all, for ELISA, for the RPPA normalization, you have all the samples on the array, right? So the normalization is straightforward. Because when I came from a genetic gene expression array background before I do the antibody-based proteomics, when I did the gene expression array, I had one sample per slide. So I have hundreds and thousands of slides I need to normalize against each other. It
Right.
generates quite a big challenge on that.
Yes, that's one of the advantages of RPPA, that you have all of your samples in one slide, and the normalization happens. For example, if you want to measure the phosphorylation of a particular set of samples, we also have a total protein that we normalize to, and then we can get the intensity values for or activation relative results for a particular sample.
That's right. So the question for Brandon is when the mass spec profiling, so normally how do you normalize the data?
So usually what you have is you have the cohort of patient, and then on the other side you have the cohort of controls, or let's say healthy individuals. The aim then will be to notice the differences that are related to the group, not to the individual. So to do so, you will pay attention on the distribution over the group. For example, if you have 10 patients, the way you will think about it is how many times can I see the proteins? Then you select the proteins that satisfy this criteria, for example, and you can do median or average protein intensity. You do the same on the control panel, and then you compare statistically the average or the mean values that you obtain for each group.
Oh,
That's
okay.
the way we basically normalized. But I will talk for myself here. When you do single cell proteomics, for example, normalization is another challenge that I will not even touch there. But I think you can do whatever you want to normalize, just so it means something and you're able to explain how you did the process. Other than that, in general, this is the way the samples are treated.
Oh, okay. So it's like the housekeeping gene normalization, right? Housekeeping protein normalization.
For us, no. We obtain individual values for each protein in each patient. There is absolutely no housekeeping gene expression that we could use as we can do on blotting method, for example, or on the technology we saw today. It's really more based on how to be strict with the data you obtain, so it makes sense and you have something normalized over the group.
Oh, all right. Thanks.
Yeah,
That makes
different.
sense. Yeah.
Yeah.
So Isela, the antibodies is the most significant and important thing over here, right? So
Yes.
how do you validate the antibodies? How do you ensure the quality of the antibodies?
So we usually just get commercially available antibodies, first of all. And we do Western blotting with positive controls and negative controls. And we look for specificity and a specific band at the right molecular weight. And that's how we decide that it's a good antibody to use for RPPA. And then we test it against our own RPPA controls and to make sure that they are behaving the way that they're supposed to be behaving on an array. So it is quite a long road, I guess, for to add a new antibody, but we do have to make sure that the data that we get from RPPA using a particular antibody that we can actually trust. So it goes to a stringent validation process before we can use it.
Okay. So for the antibodies you purchase commercially, roughly what's the validation rate? Like if you get 10 or 100 antibodies, how many can you validate the first time?
Oh my gosh. Western blotting is it's so and validation is so how do I say it? You never know what you're gonna get. Sometimes we tend to buy more antibodies from cell signaling because we can work with them they don't validate at the first time that we try to validate them. We can call them and troubleshoot, and sometimes they send us positive controls that they have used, and then we can validate it that way. um, it depends really on which antibody you're trying to validate, and if you have the correct or positive controls for it. But yeah, sometimes we can be trying to validate 10 antibodies and only 2 work the first time. So we need to do a little bit more research on the specific protocols that they are using to validate that antibody. So it's quite a consuming task, but it needs to be done.
Potential.
That's, yeah, it's a lot of work in validating antibodies. We also like the cell signaling antibodies.
Yes, and I mean, even if you keep using one antibody, the lots may change when you buy the next batch of that particular antibody, and we have to revalidate, and sometimes there are some problems, some issues, so it's not that we just validate it one time and that's good to use for forever. It also depends on the company itself. Sometimes they change lots, sometimes they change something on the antibody, and then we have to validate it again in order to keep using that particular antibody.
I see. So you have about 300 antibodies in your database. So
Around.
how do you decide what antibodies to acquire to validate?
So our research concentrate pretty much on around cancer, and depending, for example, on what type of cancer, and we do a literature search and find out what pathways may be activated or may be used in a particular type of cancer. So that's where we start. If it's a profiling of a study, then we can use up to choose 200 of them and find out which proteins are activated or not activated in a particular set of samples. So you can approach the different ways. You can select already a known set of antibodies for a particular sample that you're interested on, or you can do a complete profile and print like 300 arrays and stain for all of them, and then map the activation of a particular set.
I see. So basically, every antibody you have is in an important signaling pathway, right?
Yes.
Okay, yeah, I got it. So that's why I really like this technology. When I first started, I was thinking, okay, a few hundred antibodies, what can you possibly do? Because I came from gene expression background,
Right.
and in one array we have the whole genome. But
Yes.
I was actually very pleasantly surprised that with this 200, 300 antibodies, a lot of things could be discovered. Like when I did a gene expression array, I did the whole genome, I did projects. Not many people actually could find really meaningful changes from the whole genome. But when I did the RPPA for the labs, I mean, half of the people really find meaningful differences, and they went ahead and published the data. I was in the beginning, I think I was shocked, but I was very happy about that. That's because every single antibody is important. If you identify something, that's a whole pathway out there.
Exactly. And if you're using just one antibody that it's driving a particular pathway, then you can investigate more of the other proteins involved in that particular pathway and identify if they're downregulated or upregulated. And it's a functional work of a particular sample.
Exactly. Actually, before RPPA, many of my collaborators, they used the Western. Just guessing that pathway is important. They use Western to identify one thing. I know one lab, they are really lucky that when they guess that pathway, actually that pathway changed. But a lot of people, when they guessed it, did not change. So RPPA is a great technology you don't need to random guess, educated guess, of course. The pathway anymore, you can just do the discovery base.
Yeah, and another important thing is the amount of sample that you need for a Western. It's large volume, and for RPPA, you can print many arrays with very small amount of sample.
Exactly, that's a great point, because either Western or other technology, you really need a lot of samples. And
Yes.
for even per antibody, like for Western, you need maybe 10 or 25 microgram, but for RPPA, you can have 10, 15 microgram, you can do like 300 antibodies.
Exactly. Yes.
All right, so is there any, are there any other questions, Brandine, in the chat?
No,
Okay, so
not
we are concluding this session. Thank you again, Isela, and thank you everyone. I understand that many HUPO members may be watching the video later, the recorded one, due to time differences in those countries where it's late at night or maybe early in the morning. So I would like to extend my thanks to everyone, whether they are joining us in person today or watching the recording later. So thank you all, and thanks again, Isela.
Thank you.
Thanks, Brandine.
Bye.
See you. Bye, everyone.
Thank you very
bye-bye.
much. Bye-bye.
Bye, Giuseppe.
Thank you.